Yes, welcome back to my channel, and where we are going to study about the physiology of the cell cycle and mitosis. Physiology of cell cycle and mitosis, in which the four stages that comes: metaphase, prophase, anaphase, and telophase. First is the regulation of the mitotic chromosomes. There is a regulation of the mitotic chromosomes in which the mitotic chromosome cycle is found in the regulate by the following three control factors. First is a diffusion protein. First is the S phase activator, and that normally appear in the cytoplasm during the S phase and switch on the DNA synthesis. Rao and Johns in 1970, the M phase promoting the factors called MPF, M phase promoting factor that is present only in the M phase cytoplasm and causes the chromosome condensed. Johns and Rao in 1970. The DNA depends on the M phase delaying factors that is present in S phase cytoplasm and inhibit the process, leading the onset of MPF production. Now we study about the dissolution and the formation of the nuclear envelope during mitosis. What is mitosis? Mitosis occur when there is formation of the haploid cells. First, at least three part of the nuclear envelope that I studied, that I told you in the in the previous video, that the nucleus that enclose the nuclear envelope enclose the nucleus. It is known as the nuclear envelope, and the nuclear envelope must be considered during the breakdown of the prophase. The nuclear envelope is broke down in the prophase. Prophase, the earlier stage, is in which the nuclear envelope is get detached from it, and the nucleus is free from that. Next, and reassembly at telophase, and reassembly at in the telophase. That means the assembly is formed in the telophase stage. The nucleus having the inner nuclear membrane and outer nuclear membrane both. Then the underlying nucleus lamina and lamin protein and the nuclear pore. Having these three structures, the underlying the nuclear lamina, the nucleus having the nuclear lamina, that means the outermost layer is called lamina, and the lamin protein. The lamina is made up with the lamin protein and the nuclear pores, and there is a nuclear pores are present into it. Next, at prophase, many proteins may be phosphorylated. In prophase, the early phase, many of the proteins are the phosphorylated, having the phosphorus group in it. And then MPF. And what is MPF? Metaphase M phase promoting factor. It is known as MPF. And the phosphorylation of the nuclear lamin, which help to regulate the disintegration and reconstruction of the nuclear envelope. The phosphorylation of the lamin occur in many of the sites of the polypeptide chain and cause them the disassembles. दैट मीन्स कि जब उसके अंदर क्या होगा कि फॉस्फोराइलेशन हो रहा होगा, तो ड्यूरिंग द फॉस्फोराइलेशन there is a formation of the polypeptide chain, and those polypeptide chain are disassembled. They are not forming any assembly. They are disassembled. These are disassembled. Therefore, thereby it is disrupting the nuclear lamina. Subsequently, perhaps in the response to the different signals. See, in telophase stage. टीलोफेज स्टेज में आप ऐसा देख सकते हैं, क्योंकि प्रोफेस होता है, प्रोफेस से अर्ली प्रोफेस होता है, उसके बाद आता है मेटाफेस, मेटाफेस टीलोफेस, एनाफेस, एनाफेस के बाद लास्ट स्टेज टीलोफेस होता है, जहाँ पे कि असेंबली फॉर्मेशन नहीं होता है. नेक्स्ट working of the mitotic spindle during the anaphase. Anaphase having the is the stage where the formation of the where there is the formation of the spindles. During the anaphase, surprisingly larger force act on the chromosomes as it moves from the metaphase plate of the spindle pole. having the spindle pole. A spindle pole, that means there is a centriole, there is a centriole is present at the polar region, and from where there is a spindles are formed, and these spindle forming the plate like structure having the chromatins, and chromatins are attached with the centrioles, and when they are making the plate, this stage is known as anaphase. And then the hyd- the hydron the hydron the hydrodynamic analysis, it has been calculated that moves from the chromosome in the force of 10 to the power minus 11, यानी कि